Hello, we are going to study the understand the structure of DNA through Watson and Crick model. So DNA is a right-handed double helix structure. So basically DNA has the double helix and those double helix turn around in the right hand structure. And these contain a polydeoxyribonucleotides. That means it contains a n number of deoxyribonucleotides, and each deoxyribonucleotides are twisted around each other through the right hand helix. The bases are perpendicular to the axis. That means each of the nitrogenous bases which are present, those are perpendicular to the axis. The axis is here with a black marking line. So the bases are perpendicular to the axis. Here the sugars are, the DNA has the sugars which is deoxyribose sugar, and this deoxyribose sugar is attached to the nitrogenous bases, and these sugars are right angle to the axis. That means they make 90° to the axis. Watson and Crick model follows up with the Chargaff's rule. That means two strands are complementary to each other. That is, A is equal to T, G is equal to C. So adenine is always complementary to the thymine, but the guanine is always complementary to the cytosine. Where A plus G, that is sum of all the purines, which is adenine and guanine, is equal to the sum of the all pyrimidines, that is cytosine and thymine. So DNA strands, their nitrogenous bases are held together by two of the hydrogen bonds. Very exactly, guanine and cytosine, which is held up by the three of the hydrogenous bonds. So adenine and thymine held up together by two hydrogen bonds, where exactly cytosine and guanine held up by the three hydrogenous bonds. So the double helix structures of the DNA which are parallel to each other. So one strand moves from the 5' to 3' direction, where exactly other one moves from the 3' to 5' direction. So he can see the sugars and the nitrogenous bases over here. The nitrogenous bases contains of the guanine and cytosine, which are complementary to each other. There is two double strands here, and one is in the 5' to 3' direction, whereas another one is the 3' to 5' direction. So understanding entire mathematical logic about the DNA double helix, that is diameter of the DNA, that is approximately 20 angstrom. The diameter between the two strands of the double helix, that is 20 angstrom, where exactly if we convert into the nanometer, that is 2.0 nanometer. Single turn, so when we say something is a one single turn which is 360°, we term it as 34 angstrom, where exactly 34 angstrom contains 10 base pairs. So all 10 base pairs contribute for the one single turn, and that consists of a 34 angstrom. So understanding how much of the rotation happens in the single base pair. So that is 360 divided by 10. That single turn consists of 360 degrees, and they consist of 10 nucleotides. So that will give 36° rotation, angle rotation by each nucleotide. Same way, there is 34 angstrom with the 10 nucleotides. That will give the 3.4 angstrom. Understanding the major groove and minor groove of the DNA double helix, that is, the C1 contributes for the C1 of the ribose sugar, deoxyribose sugar contributes for the dissimilarity in the major groove and the minor groove. So what exactly happens here is the N7, the site containing N7 of purines gives the major groove, where exactly the site containing N3 of the purines gives the minor groove. The dissimilarity origin happens because of the C1 atom contributes for this. So Watson and Crick has the phosphate group, which is hydrophilic in nature. The phosphate groups contributes for the hydrophilicity, where exactly nitrogenous base contributes for the hydrophobic nature. So base pairs are the nitrogenous base pairs which are hydrophobic. So they are stacked inside where exactly the phosphate group they are present outside, so that their hydrophilicity helps in the arrangement. At the same time, the phosphodiester bonds present in the DNA acts as the backbone of the DNA.